Well, good morning, everyone. Thanks so much for joining us. I wish we could all be here together, getting to see each other. But we'll make do with this. It's a real pleasure for me to have the opportunity to introduce our keynote speaker, Dr. Rich Kowacki from St. Jude's Research Hospital. And I've actually known Rich for quite a while, since he was a grad student at Yale and I was a postdoc there. We met about 1990. And so Rich had gotten his undergraduate degree from UConn. And after he got his degree there, he actually went and worked for Boehringer Ingelheim for a number of years before he then went to Yale to do his graduate work. And interestingly enough, his very first paper was published with another Boehringer Ingelheim investigator, Steve Fezzik. So another name that we all know. And so when I met Rich at Yale, so Rich was actually in John Caradana's lab, which was around the corner from the Prestegard lab. But Rich was basically co-mentored by Jim Prestegard. And was sort of an honorary member of the group. So I saw a lot of him. I was always very impressed with Rich then because of his years in industry, Rich knew stuff about science and about life that the rest of us didn't know. And so even back then, you could sort of tell there was something special about Rich. And so Rich did a lot of his work, as I mentioned, in collaboration with the Prestegard lab. So this is a picture taken in 1998. And so Rich would have been on his way to St. Jude at that point, having finished his postdoctoral work. And this is a group of people that had trained under Jim Prestegard that just all happened to be there at the meeting together. And I was thinking back, though, of what in the Prestegard lab going on during this time period from about 1990 to 1998. What would have been going on that would sort of be identifiable as being connected with Rich? And it made me recall a very interesting sort of episode that took place right at this very time, having to do with the project that was going on in the lab when Rich would have been there. Can you advance the slide, Rich?
Thought I could.
Yeah.
Let's try. Let me get out of this. Sorry, Chuck.
That's okay.
No. How's that? There we go.
So in 1997, Joel Tolman, who was a Prestegard lab student, published a paper with Jim and his group, where what they had done was they had taken hemoglobin, and the bicell alignment method hadn't been developed yet for proteins. And so they used the paramagnet provided by the iron in myoglobin to magnetically align myoglobin, determine its structure, and study its dynamics. And what they concluded in this Nature Structural Biology paper was that the helices of myoglobin are much more dynamic than we would have guessed previously. And a very interesting thing that happened with this paper was Ad Bax was asked by the journal to write the news and views. Now, as you know, news and views are usually designed to say what an awesome paper this is. You all should read it and digest it. And what Ad did instead was he actually took the data from the Prestegard paper and reinterpreted it and said that Prestegard and co-workers were completely wrong, that the data could be interpreted that myoglobin was a very stout, non-dynamic molecule. And he had the interesting, wonderful title, Are Proteins Even Floppier Than We Thought? His answer was no. Jim Prestegard's answer was yes. And in a way, it sort of set up the field of protein dynamics going forward 20 years. And in fact, the controversy of this particular paper and its response, I contacted Jim earlier this week. It was never really resolved. Jim still stands by his results, but no one ever really did any follow-up studies to do this. But it brings up this issue of how dynamic are things in biology. So about the time this paper was published, Rich was finishing up his postdoctoral work in Peter Wright's lab. Yes, please advance, Rich. And so back one. Thank you. So Rich was finishing up his postdoctoral work in Peter Wright's lab. And somewhere around 1995, 1996, I was at an NMR meeting and met Rich and, asked him, how's your project going in Peter's Wright's lab? And Rich's response was, well, my protein doesn't fold. And back then, you couldn't cloning was really kind of slow. And so if your protein didn't fold, that was a major setback. And so I remember thinking, oh, poor Rich, his protein won't fold. This probably will be the last we ever see of Rich Krawacki because it's a disaster. And of course, advance one slide, Rich. And of course, that wasn't the case at all. Rather than viewing this as a disaster, Rich and coworkers recognized that this was a new paradigm in biology, that many proteins are natively disordered and only fold under certain circumstances, such as when they're in the presence of their cognate ligand. And so at the top here, number 3, this is Rich's invited presentation number 3. I think this was his first job talk at University of Arizona. Is protein structure a prerequisite for function? And I think the answer was no, actually. And this was actually the beginning then, and that and the paper that came out of this work was one of the events that founded the field of natively disordered proteins, which, as we all know, has become just a huge area of protein science and biology over the years. And so Rich co-founded the field. And of course, out of that field then came the phase-separated membraneless phase separation and membraneless organelles in biology. And I believe that work related to that is what Rich is going to talk about today. So, and let me just say that one of the sort of 4 things that impressed me about Rich. One is that is his biological astuteness. Rich has made major contributions to cancer biology over the years, and that's a major contribution. He's made major contributions to protein chemistry over the years. And he's done all this while maintaining a high level of expertise in NMR spectroscopy and even expanding his expertise to other biophysical methods. And so to keep all these balls in the air is obviously a lot of hard work. And so the 4th thing that really has always impressed me about Rich is his work ethic. He's a true, he's a road warrior. He goes to many different meetings, gives many talks, and obviously though works very hard to keep all his stuff, keep the trains running on time in his lab. And so I don't want to take any more of this time. Rich's talk today is going to be divided up into 3 segments, and we'll actually have a short question and answer section after each of these 3 segments. If you do have a question for Rich, I ask that you use the question answer mode, not the chat mode. I will get confused otherwise. And with that, let me just turn it over to Rich Krawacki.
Thank you very much, Chuck, for that overly praiseworthy, overly glowing introduction. It's been a pleasure to know each other for such a long time, and it's been a very fulfilling relationship over the years. And I thank the organizers of the meeting for the opportunity to present this lecture today. So as Chuck said, there are 3 parts. The first 2 are shorter than the last, and I will announce the breaks as I go along. So I'll be talking about the roles of disorder and protein function. And so first, in my lab, we use a variety of structural and biophysical methods to understand interactions between biomolecules. And in the context of the phase separation field, we try and understand how biomolecular interactions cause the formation of transient networks between molecules that then underlie the ability to undergo liquid-liquid phase separation. And we study systems in vitro as well as in cells, and ultimately, in all of our studies, we try and understand how biomolecular interactions mediate cellular function. So as I said, the talk is broken into several parts. I'll first very quickly introduce the topic of intrinsically disordered proteins. And the first vignette will discuss P27, disordered protein in cell cycle regulation. And the key concept is that P27 sort of serves as a signaling conduit in cell cycle regulation. Then the second part will focus on the protein BCLXL and its role in apoptosis. And here I want to illustrate how allosteric regulation occurs via a disordered region in BCLXL. And then the third major part of the talk will be on the topic of phase separation by a nucleolar protein, Nucleophosmon 1, which organizes the nucleolus. And then finally, in this section, I'll discuss the disordered protein ARF and how it alters the liquid phase of the nucleolus. No talk is complete without acknowledgement and thanks to everyone who did the work. So I don't have time to identify people individually. People on the left are together in a pre-COVID mode, and the newer members of the lab in an appropriately distanced manner are shown on the right. And key collaborators are identified in the lower left. And of course, none of this would be possible without sources of funding from NIH, ALSAC, St. Jude's fundraising organization, and other sources. So I thank all my coworkers very much for their tremendous contributions to our work. So I think it's well appreciated now that most proteins contain both structured domains and disordered regions. And we view these as the fundamental units of protein function. So intrinsically disordered proteins or intrinsically disordered regions of proteins are often involved in signaling and regulation. And the first two topics I'll discuss address this aspect of disordered protein function. Chuck mentioned the topic of liquid-liquid phase separation. It's now broadly appreciated that disordered proteins very commonly undergo phase separation, which drives the formation of membraneless organelles, as well as other types of biomolecular condensates. I'll focus today on the role of nucleophosmin in the nucleolus. So in particular, NMR provides atomic resolution insights into the structural and dynamic features of disordered and highly dynamic proteins. So NMR is really the go-to technique for atomic resolution insights into disordered proteins. And results from NMR complement those from other methods, and I'll illustrate our use of single molecule fluorescence, and I won't show data, but I'll discuss at least once our use of SANS in particular. We're extensive users of SANS through our collaboration with the SNS at Oak Ridge. So today's topics will illustrate different roles for disorder in protein function. So the first topic pertains to P27, which is an IDP that inhibits cyclin-dependent kinases that, when active, drive cell division in eukaryotic cells. So the complex of CDK2 on the left and cyclin A on the right with P27 is illustrated here. So P27 is about 200 amino acids, and the N-terminus is a kinase inhibitory domain which binds in the manner illustrated here in this crystal structure from Nicola Pavletich's lab in 1996. So this N-terminal region, domain 1, binds to a conserved pocket on cyclin A. Then this linking region, the LH domain, folds into a kinked alpha helix that then positions domain 2, this longer or C-terminal region, that folds into a beta hairpin, a beta strand, and a turn of helix to insert this tyrosine 88 into the active site of the kinase, potently inhibiting the kinase. But in addition, there's a 100 residue long C-terminal regulatory domain that is critical for sort of controlling the function of P27. So I want to highlight several residues that are post-translationally modified. So tyrosine 74 and 88 are in the D2 region that form the interface with CDK2, and threonine 187 needs to be phosphorylated to control the stability of P27. So there's a signaling conduit, phosphorylation-dependent signaling conduit that percolates through P27 that we were involved in discovering some years ago. So here's shown schematically the inhibited state of CDK2 with P27 bound with none of the modifiable residues modified. But through the action of several different non-receptor tyrosine kinases, tyrosine 88 can be phosphorylated, and this is associated with restoration of about 20% of CDK2 activity. And it was also known that tyrosine 74 can additionally be phosphorylated, and we recently showed that this is associated with restoration of 50% of CDK2's activity. And the significance of restoring kinase activity is that it enables threonine 187 to be phosphorylated in the context of these ternary complexes. So this is a pseudo-unimolecular phosphorylation reaction as illustrated here. And this phosphorylated threonine creates a phosphodegron for a specific ubiquitin ligase that then causes ubiquitination of P27 and lysine residues within the C-terminal tail, which leads to the degradation of P27 and the full activation of the CDK2 cyclin A complex, which drives cell division forward. So in CML, for example, the BCR-ABL fusion oncoprotein drives this phosphorylation cascade to drive hyperproliferation of those leukemia cells. And so we wanted to understand how these tyrosine residues become exposed for phosphorylation when in the crystal structure, these residues are shown intimately buried against the kinase surface. So we hypothesized that local conformational fluctuations expose tyrosine residues to solvent and to the non-receptor tyrosine kinases that phosphorylate those residues. So we used NMR, single molecule fluorescence, and biochemical methods to probe exposure of these tyrosine residues to solvent. And this was work done in collaboration between my lab with Peter Tompas in Brussels and Klaus Seidels in Dusseldorf, Germany, with other participants in the project listed on the slide. So the strategy we took with regard to single molecule fluorescence was to introduce cysteine residues near these critical tyrosines to monitor using single molecule FRET structural features and dynamic features of this region of the protein. In addition, we introduced so we used cysteine 54 as an anchor and used a second sort of point to probe at cysteine 29 that monitored fluctuations and structural features of the D1 region relative to cysteine 54. And so I'll show you sort of one piece of single molecule fluorescence data. So this is probing this 54 to 93. So this is probing conformation of Y88 in unphosphorylated P27 bound to CDK2 cyclin A. And we saw this, so this is a 2D histogram showing donor lifetime versus FRET ratio. with, on the fret scale, with long distances being at the top and short distances being at the bottom. So we saw this, sort of asymmetric pattern of fret values for this state of the protein. And if you zoom in on this 2D histogram, then you see that this peak is asymmetric, which so you can sort of see that from the projection shown in orange. And if you quantitatively analyze this 2D histogram, you can determine that there are two states that are populated, one major state to the extent of 90% and another to the extent of about 10%. And so the major state is that with a relatively short distance between the two dyes positioned here and here. So that corresponds to the conformation with Y88 in the active site of the kinase inhibiting it. But the minor state corresponds to Y88 being transiently exposed to solvent, enabling it to then be phosphorylated. And we had shown biochemically that Y88 could be phosphorylated even when bound to CDK2 cyclin A. So these single molecule fret results provide direct evidence for that exposed state of that tyrosine. In addition, we used NMR to show that phosphorylation of Y88 causes the Y88 region specifically to be ejected from the active site of the kinase. So these histograms show the chemical shift differences between the bound state of P27 and the free state of P27. So the magnitude of these bars reflect essentially the extent of folding upon binding to cyclin A CDK2. And you can see for the Y88 region, then there are chemical shift changes for this specific region when it inserts into the active site of the kinase. However, upon tyrosine 88 phosphorylation, these residues, and only these residues, are ejected from the active site of the kinase. And these are, and this is associated with restoration of about 20% of kinase activity. And you experience only about 20% activity because the rest of P27 remains bound to the kinase, and this exerts a level of inhibition that is incompletely relieved by the ejection of Y88 from the ATP binding pocket. So single molecule fret data, as well as anisotropy data, showed that phosphorylation of Y88 liberates Y74. I don't have time to show you that data. So that exposes Y74 for its subsequent phosphorylation. And NMR showed that the doubly phosphorylated P27, that this displaces the entire D2 region of P27 from CDK2, and that data is shown at the bottom. So in comparison to just Y88 being phosphorylated, the dual phosphorylation of both tyrosines displaces the entire D2 region. But note that the D1 region remains bound, still tethering P27 to the now 50% reactivated kinase, making P27 sort of a captive substrate for the activity restored by ejection of this inhibitory region of P27. So what I've illustrated to you, I'll summarize on this slide. So this inhibited state of the system sort of dynamically anticipates the structural change that's associated with phosphorylation of Y88, and this exposes this tyrosine for phosphorylation. And once that phosphorylation occurs, then this ejected state is the main conformation, but this then exposes or enables Y88 to fluctuate to conformations that expose it for phosphorylation. And then, so these two sort of multi-state, these two different two-state settings of the system are an example of sort of dynamic anticipation, is what we call it. And then upon phosphorylation of the second tyrosine, then 50% activity is restored. And this leads to phosphorylation of threonine 187 and then ubiquitination and degradation of P27. And we noticed an additional feature from the single molecule fluorescence data, and that was that as these tyrosines were sequentially phosphorylated, that the dynamics within D1 increased so as to displace D1 to a certain extent from this binding pocket. And we think that that's due to changes in the conformational ensemble sampled by this C-terminal tail and the influence that the changes due to phosphorylation have on how this region of the protein dynamically interacts with this region of the protein, leading to this displacement after both tyrosines are phosphorylated. So if there are any questions at this point, this is a break point for transitioning to the next portion of the talk.
Yeah, so a question from Matthias Buck. Hi, Matthias. The middle of P27 is not visible. What about at even higher temperature?
So yeah, that's one thing I should have noted. So the P27 CDK2 cyclin A complex is about 75 kilodaltons. So it's a pretty challenging sample to study by NMR. And unfortunately, the system is unstable and the proteins precipitate over time. So I think we performed our studies at 35 degrees as a compromise for line narrowing versus stability, but the sample basically dies after about 24 hours. So we weren't able to go to temperatures above 35 degrees, unfortunately. So the residues in the linker helix, the linker helix is actually sort of dynamic and slips and slides back and forth to a certain degree, and that's what we think is the reason for not observing the best quality sort of resonances for that region of the protein.
So Kathy Hall has a question. What is the time scale of fluctuations of P27? Could it be computationally modeled?
It could be computationally modeled. So the time scale of the fluctuations, if I recall correctly, from the single model, so Klaus Seidel actually did correlation analyses that were in the paper that we published, I guess, in 2019 on this. And the fluctuations are in the high nanosecond, low microsecond timescale range. So, a timescale that's, I guess, accessible nowadays with the computational power available now. There have been investigators that have studied the dynamics of this complex using MD in the past, but those were much shorter timescales than I think what would be relevant to the sort of the biologically relevant fluctuations that I've discussed here.
Rich, I have a question. So when threonine 187 gets phosphorylated, you said that that turns it into that becomes a degron that leads to degradation. Is that because of dissociation? Is that what the degradation system sees, or is there actually a conformational change that gets recognized by a ligase?
No. So I mean, that's one of the sort of beauties of this system is the threonine 187 is basically dangling at the end of this highly flexible C-terminal tail. And so it's very accessible for the E3 machinery to come in and ubiquitinate lysines within this tail. And then after polyubiquitination, then the it becomes a substrate for the 26S proteasome. And so then the proteasome basically pulls P27, into this proteolytic machine and pulls P27 off CDK2 cyclin A, releasing them to be fully active.
I see. Great. So why don't we move on, Rich, to the next segment of your talk?
Okay, here we go. I don't know if I could. Okay, so in this next section of the talk, I'll talk about BCL-XL and apoptosis, and again, allosteric regulation via a disordered region. So this was work done by a former postdoc, Ariella Folis, in collaboration with my close colleague, Doug Green, in partnership with a number of other individuals, including Francesca Marassi at the Sanford Burnham and others in Doug's lab and my lab. And so this pertains to regulation of apoptosis by P53. So everyone is probably aware of that the P53 tumor suppressor has nuclear functions in response to, for example, DNA damage. One of P53's transcriptional targets is a BH3-only protein called PUMA, but it's less well appreciated that P53 also has cytosolic roles under stress conditions. So some P53 is diverted to the cytosol where it has the potential to directly activate the pro-apoptotic protein BAX. But at first, it is bound and inhibited by BCL-XL. But when PUMA is also expressed, PUMA binds to this binary complex and actually displaces P53, enabling P53 to engage BAX to cause structural changes in BAX that cause its oligomerization. This permeabilizes the outer mitochondrial membrane, releasing cytochrome C, and through a sequence of steps triggering apoptosis. So Ariella had a good run in the lab, and he sort of described the molecular basis of several of these key signaling steps in this pathway. What I want to discuss today are his studies of the P53, BCL-XL, and BH3 domain protein complex. So BCL2 inhibits apoptosis by binding and inhibiting P53 as I've described, but also by binding through a separate interface the BH3 domains of pro-apoptotic proteins other than the protein PUMA shown on the last slide. And so I want to tell you about what we sort of learned about how these interactions are regulated by a disordered loop in BCL-XL. So shown here are the schematics of the structures of several BCL2 family members, pro-apoptotic members at the top, anti-apoptotic members at the bottom. And what's noteworthy at the bottom is that BCL2 and BCL-XL each have long disordered regions in between helices 1 and 2 within the structure. And the sequence of this IDR for BCL-XL is shown here. And it was known from the work of others prior to our studies that there were several residues that were post-translationally modified. And this was associated with a reduction in the anti-apoptotic activity of BCL-XL. So this loop is shown here on the right in the context of the folded structure of BCL-XL bound to a BH3 domain. So as I said, it's about a 60 residue long loop between helices 1 and 2 with several post-translationally modifiable sites. Many of them are kinase phosphorylation sites. And so one of these sites in particular, serine 62, was known, when it was phosphorylated, it was known that this attenuated the anti-apoptotic activity of BCL-XL. So Ariella wanted to understand how this change in anti-apoptotic activity occurred due to the phosphorylation of a serine residue kind of in the middle of this flexible loop. Interestingly, this loop was cut out of proteins that were initially studied in structural studies. For example, the first NMR structure by Steve Fezzik actually sort of omitted this loop, and there were several subsequent crystal structures that omitted this loop. But the loop is actually a very important part of BCL-XL that allows its function to be regulated. So shown here are results for comparing wild-type BCL-XL with a phosphomimetic form of it in which serine 62 is replaced by a glutamate. And you can see, you would sort of expect that, okay, you change one residue in a disordered loop, not going to see very many chemical shift changes in the folded core of the protein. But in fact, you see changes kind of throughout the protein, which was really intriguing. So it turns out that this phosphorylation of this residue causes interactions with this region of the folded core of BCL-XL, which is where it binds to p53. But then additional structural changes occur in the BH3 binding groove of BCL-XL. So there's sort of allosteric between direct interactions in this part of the protein through the core of the protein to the BH3 binding domain. That, I should have mentioned, that importantly decrease the affinity for BH3 domains of pro-apoptotic proteins that are being inhibited by being bound by BCL-XL by a factor of 5 or more. So Ariella solved the structure of this phosphomimetic form of BCL-XL, and he showed, as I mentioned earlier, that this negatively charged phosphoserine interacts with a patch of arginine residues. One in particular, arginine 103, is sort of the center of this arginine-rich patch. And this interaction then alters the conformation of helix 3, which is adjacent to the BH3 binding groove, which is located here. And these, located here. And so this creates a steric clash at the base of the BH3 binding groove. And this is illustrated more clearly on the left. So these multiple structures that are overlapped here are sort of in the absence of the phosphoserine when bound to BH3 domains. And but when the serine is phosphorylated, this helix 3 shifts to the left, which pinches off the bottom of the BH3 binding groove, which is then associated with an increase in the KD value or a decrease in affinity for the pro-apoptotic proteins. And so this is a mechanism for releasing those pro-apoptotic proteins to engage BAX and BAC and other pro-apoptotic effector proteins. So what I've illustrated to you then is that upon modification of the BCL-XL IDR, that I didn't emphasize this, and I need to, it binds to the same surface as p53. So this then inhibits p53 binding and essentially releases p53. And then through this allosteric mechanism, the BH3 binding groove is altered, and that causes release of BH3 domains. So there are sort of two dual mechanisms of promoting apoptosis via this serine phosphorylation, the release of p53 and the release of BH3 domain proteins to promote apoptosis in cells. And in the paper we published on this, I guess in 2018 it was, there was extensive cellular data supporting this mechanistic model. So to summarize what I've told you in this portion of the talk, BCL-XL can engage BH3, proapoptotic BH3 domain containing proteins, as well as P53. One mechanism that we described that promotes apoptosis that I haven't gone into, that was a part of our early studies, is that the BH3 protein PUMA can bind to this BH3 groove, releasing the directly bound BH3 proteins, but it also, through an allosteric mechanism, disrupts helix 3 and the interface with P53, leading to both the release of a BH3 protein as well as P53. And what I told you about in some detail just now is how phosphorylation of a residue in the loop directly causes P53 release through direct competition with this interface, and then how through an allosteric mechanism BH3 domains are released. And just one final point is, so essentially these two pathways utilize, or these two mechanisms for engaging apoptosis via BCL-XL engage the same allosteric pathway in the protein, but in opposite directions. This one from BH3 to P53 surface, and this one from the P53 surface to the BH3 binding groove. So this is a breakpoint for the second part of the talk. If there are any questions, I actually have the question window open, so there are no questions posed, at least so far. If anybody has a question on the panel, feel free to ask. Otherwise, I can proceed. I guess this was...
Richard, I have a quick question. So that flexible loop in the bundle, in the structure bundle, looks like it is fairly fixed. It's a fixed structure. So how flexible is that loop actually, or what stabilizes it?
So Ariella, so there are electrostatic interactions between the phosphoserine and arginine residues on this surface of BCL-XL in the folded core. And so Ariella was able to observe interresidue NOEs between the loop and the folded core in this region of the loop. Other portions of the loop there were more flexible, and there were not sort of loop folded core NOEs. But he was able to get a sufficient number of restraints, mostly NOE restraints, in order to describe the conformation for this portion of the loop.
It's more like a stabilized loop.
Yeah, it's like a stabilized loop, but there's extensive data using phosphopeptides and loop deleted BCL-XL in the paper, showing that, I forget the exact value of the affinity constant or the dissociation constant, but it's in the, I think it's in the sort of low micromolar range when you study the interaction using peptides and the folded core of BCL-XL. But in the context of the loop within the protein, then it's a strong enough interaction to give rise to NOEs. So yeah, it's a stabilized loop, I guess, would be a good way to describe it.
Thank you.
Rich, there's a question from Catherine Brewer, a grad student from my lab. She asks, have you done any experiments to explore P53 competition with the IDR loop? Is there a certain concentration of P53 that can feed off this loop?
Yes, I forget the details, but that data is in the paper. Yeah, so we were, because we had previously described the P53 BCL-XL complex, And so then Ariella was able to disrupt that complex by titrating in a peptide corresponding to the central portion of the BCL-XL loop. So that was, yeah, very good question, and we were able to demonstrate that. Okay, I guess I will move on. Oh, yeah, so I'd like to summarize this portion of the talk. So the IDP-P27 functions as a signaling conduit via multi-site phosphorylation. So this is an illustration of, you know, how disorder within a protein can participate in transduction of sequential phosphorylation signals. And the second part shows that the internal loop or IDR of BCL-XL allosterically regulates anti-apoptotic function via post-translational modification. And an important point is that there are additional modification sites within that loop. So you can think of the loop as being sort of an integrator of cell signaling. And, you know, maybe it's quite possible that multiple modifications could occur via the action of, you know, different signaling kinases. And that would probably amplify the allosteric effects and you know, and basically further reduce anti-apoptotic activity. So it's a way of, you know, in a graded way, controlling BCL-XL's anti-apoptotic function. So now I'd like to transition to the final section of the talk regarding phase separation. So specifically phase separation by Nucleophosmon, which organizes the nucleolus. So I imagine that many of you are now familiar with the fact that there are many membraneless organelles in cells that form through the process of liquid-liquid phase separation. And the largest of these, and actually the one that's been known to exist for the longest time, is the nucleolus, which is comprised of ribosomal, the genes that encode ribosomal RNAs, the ribosomal RNAs themselves, and ribosomal and non-ribosomal proteins. And it's the center for ribosome biogenesis as well as cellular stress signaling. So just a very quick introduction to the topic of phase separation. So phase separation is driven by weak and transient interactions between multivalent biomolecules, be they proteins, DNA, RNA, et cetera. It's concentration, temperature, and ionic strength dependent, and therefore can be modulated by post-translational modifications. It occurs through the formation of transient non-covalent networks between biomolecules, as I illustrated in my introductory slide, and it organizes biomolecules within cellular compartments to perform diverse biological functions, including ribosome biogenesis, transcription, and many, many other biological processes. So nucleophosmin is the major constituent of what's called the granular component region of the nucleolus, where it mediates the assembly of pre-ribosomal particles. So shown on the upper left is an image from Cliff Brangwen's work of an actual nucleolus, showing that it's compartmentalized into several different regions. And so each of these green regions as a multi-layered structure that's illustrated schematically in the center of the slide. So at the very center of those green regions are what are called the fibrillar centers, which is where the genes encoding ribosomal RNAs are clustered together. And those genes are transcribed to produce ribosomal RNAs, which then are captured through phase separation with the protein fibrillarin, creating this second layer in which those RNAs are spliced and modified. And due to constant transcription, then there's a flux of mature RNAs outwards, and the RNA that emerges from the middle layer, the DFC, then is captured through phase separation with the protein nucleophosmin, creating this outer granular component region of the nucleolus. However, at the same time that RNA is moving outwards and phase separating with nucleophosmin, ribosomal proteins are being made in the cytosol, are transported through the nuclear pore into the nucleoplasm, and then they interact and phase separate with nucleophosmin in this granular component region, creating sort of an incubator then for the assembly of those ribosomal proteins with the ribosomal RNAs to create ribosomal subunits. So what role does nucleophosmin play in the nucleolus? Well, I already said what its role is. Its role is to drive phase separation with ribosomal components. So shown here is a video of what happens when nucleophosmin is mixed with a single ribosomal protein, RPL5. So neither of these proteins will phase separate under these conditions by themselves, but when they're mixed together, they spontaneously form these droplets that you can see fuse rapidly. The components within them are highly dynamic based on FRAP analyses, and the droplets are dense and viscous. So the nucleophosmin concentration within these droplets is about 4 millimolar, giving you insight into sort of the ability of phase separation to concentrate molecules within these liquid environments. And the droplets are quite viscous, roughly that of thick honey. So how does nucleophosmin do this? So it's a pentameric protein. So at its N-terminus is a pentamerization domain that creates extensive multivalency of the other domains of the protein. At the C-terminus is a small helical DNA and RNA binding domain that mediates interactions with ribosomal RNA. And then connecting those two folded domains is an intrinsically disordered region of about 120 residues. And prominently, the IDR has two acidic tracks. And these acidic tracks, so the IDR is, of course, displayed 5 times. And then there are, in total, 10 acidic tracks within this region of the protein. And these acidic tracks interact transiently with clusters of arginine residues in other proteins, ribosomal proteins and other nucleolar proteins, via electrostatic interactions. So the charge density of the IDR of nucleophosmin is shown here with the 2 acidic tracks, and these acidic tracks then interact with clusters of arginine residues and other proteins. So this is the charge density plot for RPL5. And so you can think that these clusters of arginines interact transiently with these acidic tracks, and then this other arginine cluster can interact with an acidic track in a different nucleophosmin pentamer. And so when the concentrations are appropriate, then you establish sort of networks of non-covalent crosslinks driven by electrostatic interactions between the molecules, and that's the molecular basis of phase separation. So nucleophosmin is particularly interesting in the realm of phase separation because it can phase separate through 3 independent mechanisms. One is with ribosomal RNA, as I've already mentioned. This is a heterotypic mechanism of phase separation, schematically illustrated here. The second that I've illustrated to you is heterotypic phase separation with either ribosomal proteins or non-ribosomal proteins that contain multiple arginine motifs. And then we also described that nucleophosmin can phase separate with itself, homotypic phase separation under crowded conditions, for example, as exist within the nucleolus. So how do these different mechanisms of phase separation contribute to ribosome biogenesis? So in 2018, we published a sort of a model for how we think nucleophosmin mediates ribosome subunit assembly. And very recently, together with Cliff Brangwyn, provided experimental support for that model. So the model goes like this. So here's a mini nucleolus at the top of the diagram, and here's the a granular component region, which is expanded below. And so we have ribosomal RNA being produced in the interior, moving outwards. So that RNA can instantaneously phase separate with nucleophosmin in this region of the nucleolus. At the same time, we have ribosomal proteins coming in this direction into the nucleolus. So we have sort of countercurrent fluxes of RNA in this direction and ribosomal proteins in this direction. And so the ribosomal protein, shown as these blue squiggly lines, can then interact with the ribosomal RNAs, forming ribosomal subunits. And when this occurs, then it buries the interaction sites of these ribosomal components that were previously interacting with nucleophosmin. So that weakens their interactions with nucleophosmin, allowing these assembled subunits to passively diffuse out of the nucleolus. And then shown here are sort of homotypic interactions between nucleophosmin molecules that we think sort of sustain the liquid nature of the nucleolus when ribosomal subunits are essentially breaking away from nucleophosmin through their assembly. And so now I'd like to wrap up and summarize. So P27 is a disordered phosphorylation-dependent signaling conduit that regulates cyclin-dependent kinases and cell division. And so the phosphorylations percolate through these tyrosine residues, activating the kinase, phosphorylating the threonine, leading to ubiquitination and degradation. And then I told you how phosphorylation of a loop in BCL-XL decreases anti-apoptotic activity, leading to the release of two types of pro-apoptotic effector molecules. And then I've told you about how disorder within nucleophosmin mediates phase separation with ribosomal components. And coming back to the theme of NMR, NMR has been important for us in all of these studies. But in addition, complementary biophysical and structural methods have also been important. Single molecule fluorescence in the case of this system, and SANS in the case of studies of nucleophosmin, as well as other systems under study in the lab. So that leads me to thank once again lab members past and present, funders, as well as collaborators, to thank the audience for your attention, and I'll of course be happy to take any questions there may be.
Thanks, Rich. Clearly, proteins are in fact more floppy than we thought 20 years ago. So here's a question from an anonymous attendee. Maybe I misunderstood. Looked like there was a fourth R-rich region in the diagram, or was it something else?
No, that was a fourth arginine-rich region. I think it didn't rise to the level, or I think it doesn't rise to the level of charge density to be called a bona fide ARG motif via our probably somewhat qualitative naming system. But clearly there are additional arginines in the very C-terminus. So yeah, good call on that.
So a grad student from my lab, James Hutchison, asks, if you make a ternary mixture of NPM1, P14ARP, and ribosomal RNA, do you think you would see both glassy and liquid diffusion in separate domains, or a mean diffusion between the two types of NPM1 interactions?
Oh, that's a very good question. We have not progressed with our studies of ARF to also include ribosomal RNA in the in vitro droplets. We do in other studies. There are several members of the lab, in particular, Michelle Tolbert, shown in the back of the pre-COVID together photo. Michelle is an RNA expert, and isolating ribosomal RNA in the lab and studying its interactions with nucleophosmon and influences of other ribosomal proteins, and in addition, what happens when ribosomal proteins are added. But we haven't yet sort of additionally introduced ARF into that more complex type of condensate system. But of course, ribosomal RNA is present in cells, and so our ongoing sort of imaging studies into the dynamics of ARF and nucleophosmin in the nucleolus will be probing those behaviors in the context of nucleoli, which contain tons of ribosomal RNA. So yeah, thanks for that question. I'm sorry I can't be more specific with my answer.
So I think probably we better wrap it up and turn it back over to Marcus. Rich, that was an absolutely fantastic talk, and if we're a little over in time, it's only because of my long-winded introduction. So Marcus, turning it over to you.
But it was a fantastic morning session. I would like to thank all the speakers that contributed to this morning for their talks, and it was clearly stimulating with all the questions we had. So fantastic session this morning. Thank you, all the speakers.